It's been more than 25 years since HIV, the virus that causes AIDS, was first discovered.
It is the disease of our generation. It changed the fate of everyone, I think, in the globe.
And yet, scientists are not any closer to finding a way to prevent AIDS. They have found it to be a highly elusive target.
So a virus which is constantly changing, constantly provide a constant moving target to the immune system. The immune system cannot keep up with the virus. As soon as you elicit an immune response against one virus, the population within your body is completely changed.
Safrir Moore is a researcher at ASU's Biodesign Institute. He leads a team that is working on a research project to produce a vaccine to prevent the infection of HIV. For clues on how to make their vaccine, they've looked at people that seem to have natural protection against HIV infection.
Our strategy is to, again, focus on those lucky few that manage to avoid being infected by the virus despite the fact that they are exposed to it. So their body somehow knows how to potentially get rid of it. In order for us to create a vaccine, we need to establish what are the correlates of protection, what in the protected person, naturally protected person, what gives it the edge over the virus.
They are focusing their work on an HIV protein that helps the virus first enter the body, called GP41.
They have antibodies that are directed against one of the proteins of the virus, one of the envelope proteins of the virus, not the one that was mostly studied in the last 25 years. It's not the GP120 portion, but it's the part which anchors the GP120 portion of the envelope to the membrane of the virus itself. It's called GP41.
The goal is to use GP41 as the basis for a new vaccine to prevent the initial HIV infection and stop the spread of AIDS.
These antibodies, when tested in an ex vivo system for looking at how the virus is penetrating through the mucosal barrier, is able to block, those antibodies are able to block the virus from going through the epithelial cells.
To produce large amounts of the vaccine at a low cost, they have recruited an unlikely ally, tobacco.
Our goal is to produce this particular protein in high levels in plants, and that goal was accomplished. We can now express it, we can extract the protein, clean it, and use it for immunization.
The overall goal of this project is to produce HIV proteins in tobacco, and we do this through a process called stable transformation, in which we take bacteria that harbor the genes for HIV and we put those with the tobacco, and then the proteins are produced. So this plate is 1 day after stable transformation, so you really can't see anything. The plant looks fairly natural right now. Then 2 weeks after transformation, we have very little calli starting to form, which these are the stably transformed cells. Those cells then start to form these calli, which are the darker green parts in this tub right here. And those calli will eventually differentiate into shoots, which we have. This is approximately 6 weeks after stable transformation. We will then take those shoots and put them in rooting media and eventually have whole plants, which we will regenerate into seeds, and then we'll have stably transformed tobacco that produces our HIV proteins.
Finally, the team can begin to harvest the HIV proteins from the tobacco.
So what we're looking at here is the difference between a wild type that has not been infiltrated and a plant that has been infiltrated 1 week post-infiltration. So here we have the wild type that's nice and healthy and green, and the infiltrated plant is fairly necrotic, and that's basically wherever the protein is being produced is that necrosis. So just like I said, non-infiltrated 1 week post-infiltration.
Moore's group has demonstrated success, and they hope to continue their research to one day begin clinical trials.